this next chapter, we will build on the chemical level of organization by looking at the cellular level of organization. There are two types of human cells. There are sex cells, otherwise known as gametes, or germ cells. These gametes are sperm and oocytes, and the job of these cells is to make new humans. The second cell type is the somatic cells. These cells make up all the other body cells. A cell has a plasma membrane that surrounds it. It's the cell's boundary from other cells. It separates the extracellular environment, or what's outside of the cell, from the intracellular environment, what's inside the cell. The plasma membrane helps to keep the conditions inside the cell different from the conditions outside of the cell. We call this feature of the plasma membrane selectively permeable, meaning it lets some things into the cell while keeping other things out. Inside the cell, there are membranes that surround some organelles. Examples include membranes that surround the cell's nucleus, mitochondria, and the Golgi apparatus. The inside of the cell is called the cytoplasm and is separated into two components. The cytosol is the liquid part, and the organelles are the intracellular structures that make up the second part. I want you to spend some time reviewing the diagram of the cell in your lecture guide and start familiarizing yourself with the different organelles. We will discuss each of these organelles in greater detail later in this chapter. Let's talk about how cells grow and are made. All cells come from the division of pre-existing cells. Most cells are divided by mitosis, which results in the production of two identical daughter cells. To understand how these cells are produced, we need to take a closer look at the DNA, which is in the nucleus of the cell. The nucleus of the cell is the control center or brains of the cell. Most cells in our body have a single nucleus. There are some exceptions. For example, red blood cells do not have a nucleus, and skeletal muscle cells have many nuclei. The nucleus is surrounded by a nuclear envelope that separates it from the cytosol. Inside the nucleus, we have a nucleolus, which is a transient nuclear organelle, which means that sometimes it's there, sometimes it's not, depending on what the cell is doing. The nucleolus synthesizes ribosomal RNA. The nucleoplasm is all the fluid between the nucleolus and the nuclear membrane. It consists of a nuclear matrix. The nuclear matrix provides support to the nucleus. Inside the nucleus is the most important thing, DNA. DNA holds the genetic material and is organized in the nucleus as coils. It's coiled around a histone protein. DNA wrapped around a single histone is called a nucleosome. DNA is a huge molecule. For it to be able to fit inside the nucleus, we must wrap it around these histones. DNA plus the histone proteins is what makes a chromosome. Chromosomes take on two conformations or structures depending upon whether the cell is dividing or not dividing. In a non-dividing cell, it's in loose coils called chromatin. Under a microscope, chromatin looks like a mass inside the nucleus. When the cell is entering the early stages of cell division, the chromatin will condense to form visible chromosomes. Packing the DNA into chromosomes makes it easier to separate the DNA during cell division. Let's talk about the phases of the cell cycle. The whole idea of this is to produce two daughter cells, each about half the size of the parent and genetically identical. The cell cycle is divided into two phases. The mitotic phase is known as the M phase and interphase. The M phase is further divided into mitosis and cytokinesis. Mitosis refers to the division of the nucleus. This occurs as the cell splits its duplicated chromosomes into two cells. The division of the cytoplasm, which occurs near the end of mitosis, will create the two daughter cells and is referred to as cytokinesis. Phase is the phase where the cell spends most of its time. During interphase, the cell is carrying out its normal function. When it's ready to divide, the cell will enter gap 1. Gap 1, or G1, is where the cell replicates its organelles and increases the volume of the cytosol. From there, the cell moves into the S, or synthesis phase. In this phase, DNA duplication is performed, and at the end of this phase, the cell has two identical copies of its DNA. The last and final phase before mitosis is called the gap 2, or G2 phase. During this phase, the cell will replicate its division machinery and make proteins necessary for the division process as it prepares to enter mitosis. If we were to compare the cell cycle to a 24-hour clock, the cell would spend about 23 hours in interphase and the final hour in mitosis. The rate of cell division, where a cell produces two daughter cells, varies greatly based on cell type. So it's important to understand that the time in interphase is really what decides the rate of cellular division. The process of mitosis takes a precise amount of time, between 1 to 3 hours from the time it begins until two separate daughter cells are formed. This means if a cell spends several days in interphase before it divides, its rate of cell division is slow. While if a cell spends 15 hours in interphase before it divides, its rate of cell division is very fast. Because mitosis produces two genetically identical daughter cells, the cell must make sure that each cell has the same amount of DNA. The process for DNA replication occurs during S phase, as previously mentioned. There are several enzymes involved in this process: DNA polymerase, helicase, and ligase. For DNA replication to occur, the first thing that needs to happen is the DNA needs to unwind. The enzyme that does this is called a helicase. Helicase gets its name because it unwinds the DNA helix. Once the helix begins to unwind, there is room for the DNA polymerase enzyme to attach itself to the strands of DNA. The DNA polymerase replicates each strand of the DNA by attaching complementary DNA nucleotides. While not seen in the figure, multiple polymerase enzymes will bind to the DNA. As the enzyme moves down the DNA, they will copy it. DNA polymerase moves only in one direction down the DNA strand, so one complementary strand is produced in a continuous fashion. The other strand is produced in fragments. These fragments are then joined together by the ligase enzyme to create one continuous strand. In the end, we have produced two identical copies of the original DNA. To divide the cell, we continue in two phases: mitosis and cytokinesis. At the end of interphase, the DNA exists as chromatin, a thread-like mass. The first phase of mitosis is prophase. During prophase, the chromatin will condense to form chromosomes. The nuclear envelope and the nucleolus will disappear, and the centrioles, which are the machinery needed to move the chromosomes during the division, will migrate to opposite poles and form what are known as spindle fibers. Next up is metaphase. During this phase, the chromosomes will align along the equator or the center part of the cell. This cell has a total of 4 chromosomes. So we have 2 red chromosomes and 2 blue chromosomes. The structure of a chromosome consists of 2 sister chromatids, which are joined together at the centromere. After metaphase comes anaphase. During anaphase, the chromosome and the centromere split in half. So each sister chromatid is now pulled to the opposite sides of the cell. Chromosome movement will stop after arriving near the centrioles at the opposite poles of the cell. From here, we enter telophase. Telophase marks the end of mitosis, and in this case, nuclear division is now over, and the nuclear membrane will reform. The chromosomes will uncoil to form chromatin. During telophase, cytokinesis will begin with a little indentation that forms. This is called the cleavage furrow. Cytokinesis will continue until the division is complete and 2 separate daughter cells are formed. This marks the end of cell division.